And what I'm going to do over the next 25 minutes or so is really give you an introduction and some highlights as to what we've been doing as a research group in London over the last 15 years or so now at high altitude, and how that relates to the patients that we look after in an intensive care unit at sea level. So I have a couple of disclosures at first, as we all do these days, but I think perhaps the most important disclosure is that I've been really very hypoxemic at high altitude before, so this may have either caused some damage to my brain or give me a slightly unusual view about hypoxia and hypoxemia and how we should deal with patients with these conditions. So I'm sure that most of you are familiar with what an intensive care unit looks like. We look after the most unwell patients in the hospital, most of whom are suffering from multiple organ failure. And what is common amongst these patients is that almost all of them suffer from hypoxemia, a lack of oxygen in the arterial blood, which can lead to hypoxia, a lack of oxygen at a tissue level. But what's been very difficult over the years is trying to study these patients, understand what it is that is affecting them so badly, and trying to develop ways of improving their outcomes. As I'm sure those of you with a scientific background understand, what we try to do in science is design an experiment where you change one factor and one factor alone, and then try and evaluate the impact of that one factor. The problem with intensive care patients is that they do not come like this. Our patients are a very heterogeneous group of patients doing a ward round in the morning on an intensive care unit. This is what a sort of list of diagnoses may look like. And this is perhaps why over the years we've been quite bad as a specialty at trying to help our patients, and it's just because they have so many different underlying pathologies. So what we've been doing over the last 15 years or so is developing a model, if you like, a new paradigm to try and understand the critically ill patient, which is to take healthy volunteers to high altitude, understand what is advantageous and disadvantageous to them from the perspective of biological processes, and bring that information back to sea level, back to the intensive care patients to see if we can improve the outcome of the patients that we're looking after. So this is, if you like, a conceptual diagram of really what we're trying to do is taking these healthy people to the mountain, looking at different biological, genetic, and biomarker information, seeing what helps people adapt well and less altitude, and then bringing that back to sea level, and then undertaking both observational and interventional trials to improve their outcomes. Over the time that we've been doing this, we have looked at an enormous number of different biological processes, trying to understand more and more about adaptation to low levels of oxygen. And I'm clearly not going to have time to talk about all of the things that we've looked at during this short talk, but I'll try and touch upon some of the more important ones, perhaps, just to really focus down on what we think might be going on both at altitude and in our intensive care patients alike. Now, you might think this whole model is perhaps just a reason to get us out on the mountains. Perhaps that may be true to some extent, but its true use as a scientific model, actually, when you think about it, although it has clear limitations, the other models that we use in science today also have pretty large limitations. So whilst we may understand a lot about what's happening in the human body from small furry animals, mice and rats, and both cells in Petri dishes, these techniques are very, very limited in being able to tell us what's happening in complex, multi-system human diseases. So we think that our model is not perhaps as bad as some make out to be. So I'm going to skip through the next bit quite quickly with this audience, because I think you probably have a good understanding of the physiology of high altitude. But this is just simply to remind you that as we ascend to high altitude, there's an exponential fall in the barometric pressure as you get higher and higher. And what this leads to is a reduction in the partial pressure of oxygen that you breathe. So the concentration is the same, the barometric pressure falls, and the combination of those two means that there's a reduced partial pressure of oxygen. When you get to the base camp at about 5,300 meters, the partial pressure of oxygen is roughly half of what it is at sea level. And if you get to the top of the summit of Mount Everest, the partial pressure of oxygen is about a third of that you would see at sea level. So really very low levels of oxygen that we're looking at in the experimental model that we've created. The effect that that has on your physiology is fairly profound. So the oxygen cascade that we talk about, this waterfall type effect of a reduction of the partial pressure of oxygen moving from the atmosphere through the different convective and diffusive systems in our body, right down to the level of the mitochondria that uses the oxygen, which we'll come back to later. All of this is dramatically affected by the reduction of partial pressure at the top end. And the ultimate effect is hypoxemia. I took these pictures recently. Heimo may recognize them. We were recently at a hypoxia meeting in Atacama Desert. And these telescopes are about 5,000 meters or so. And here is two of us with these oxygen saturation probes. And you can see the other thing that is also fascinating about high-altitude medicine, which is very similar to what we see in the critical care unit, is that you can take two people who physiologically look very similar, and you can put an oxygen saturation probe on at the same place, and you can see one of them reads 66 and one of them reads 86. And this is just the same as we see in the intensive care unit, where you can't really predict how well or badly people are going to do in any given situation based on what you see in front of you. So, over the last nearly 15 years or so now, we have undertaken a number of expeditions to high altitude to try and understand more and more about what I've been explaining to you. I'm not going to be able to explain all of these expeditions in detail. In 2007, this was the largest of our expeditions. We took 220 members of the public to Everest Base Camp, testing them at various altitudes on the way up to base camp. And a further 14 of us were lucky enough to climb on the mountain and continue the research all the way up to the summit of Mount Everest and back down again, spending a total of around 65 days at altitude testing each other along the way. This is, I'm sure most of you, or many of you at least, have been to Everest Base Camp. This is sort of what it looks like in the midst of the climbing season. There can be anything up to 1,000 people or so living at base camp, very busy place. These here at the front are our scientific research laboratories in 2007. And this is the sort of setup we tried to build inside the laboratories. So fairly similar to what you might expect at a laboratory at sea level, with fairly sophisticated equipment that is a fair struggle to get working at these sorts of altitudes. We carried out an enormous number of experiments on this particular expedition. Here you can just see a few of the things, some neurocognition testing. This is looking at the back of the retina with a digital retinal camera, and lots of exercise physiology during that expedition. I just want to share one part of that expedition with you, just one small experiment that we conducted, and we were interested to try and measure the level of hypoxemia in climbers sat on the top of Mount Everest. So we took femoral blood gases from one another from base camp all the way, hopefully, to the summit and back down again. This is, some of you may recognize, this is the South Col of Mount Everest, and this is where we leave to go to the summit of Everest. In the foreground here is our camp. In this tent at the front is an exercise bike where we were doing VO2 max testing at 8,000 meters on one another, and the next morning we left to leave for the summit. We were lucky enough to have a fairly good day for climbing and summited on the 23rd of May, 2007. The problem was that it was a very windy day. maybe 25 miles an hour of wind on the summit. And with the femoral blood gases, we needed to take off our down suits and strip to our underwear. So we decided that it was perhaps a little bit dangerous for that day and descended down to 8,400 meters to an area called the balcony, where we put up a small shelter, took blood gases from one another, and brought the samples back down to Camp 2 to measure them. Here we had a benchtop analyzer like you might find in an intensive care unit or operating theater, and measures the samples as they came back down. You may have seen the data before, so I'll just quickly summarize it. 4 blood gases, one from each of us that had blood gases taken from us. Here you can see the individual values in millimeters of mercury, with the average partial pressure of oxygen of those climbers as 3.28 kilopascals, which gives a calculated oxygen saturation of 54%. This person here, subject 2, particularly low levels of oxygen, a partial pressure of oxygen of 2.54 kilopascals, which gives a calculated oxygen saturation of 34%. This was my blood gas at the time, not necessarily the lowest, but one of the lowest arterial oxygenations ever recorded in a human being, and I'll leave you to judge the effect that that's had on my own neurocognition since then. That experiment was all very well. It doesn't prove anything scientifically other than the fact that some human beings can tolerate very low levels of oxygen without any obvious harm to them. But the real question being asked here is how we are able to do that. Why is it that some humans are able to adapt to these very low levels of oxygen and others are not? We've probably published somewhere in the region of 35 to 40 papers now trying to explain our ideas about the various different mechanisms that we believe to be important in that process. And I'm just going to pick out 1 or 2 of those areas now to try and describe what we think leads to these, the ability to adapt to these very low levels of oxygen. I think you're all probably very familiar with this classic description of acclimatization to high altitude, so I'm not going to dwell on it for very long. But most of it really describes an augmentation of systemic convective oxygen delivery. So you breathe harder and faster, your heart rate increases, your cardiac output increases, your hemoglobin increases, and you just deliver more oxygen to the tissues. This is a very nice story, but it fails to explain the differences between people in terms of performance and survival, both on the mountain and on the intensive care unit. So what we've tried to do is look further along that oxygen cascade that I showed you and look right at the end of the oxygen cascade into how blood and oxygen is delivered on a microscopic scale, and then how oxygen is used at a cellular and mitochondrial level to see if that can show us any differences between individuals. So what we've been spending a lot of time looking at now is the microcirculation, mitochondrial function, and how nitric oxide may control that component of tissue local oxygen delivery and consumption. Another big part of the story is these guys. And when we came back from Everest in 2007, giving talks like this, so many people said to us, "But what did you find in the Sherpas?" And unfortunately, in 2007, we were not able to study the Sherpas for many reasons. The main reason was we hadn't thought about it, but then also we didn't have ethical permission, and they were kind of busy looking after us. So in 2013, we went back to Everest and conducted another large-scale expedition with 180 people this time we were studying, but this time 65 of them were Sherpas. And for any of you who have had the joy of meeting these Sherpas in Nepal, I think you'll agree with me that their ability to withstand low levels of oxygen are truly astounding. So they perform on the mountain as if they were at sea level, and there's very little is understood about how it is that they could actually do this. So those factors that I described earlier about oxygen delivery, so heart rate, cardiac output, hemoglobin, lung function, none of those are different between us and the Sherpas. So those simple things do not explain the differences between us and Sherpas. So we returned and basically did exactly the same thing as we did in 2007, but this time we studied us against the Sherpas, and we kind of held the Sherpas back to our very slow rate of ascent, and they walked alongside us all the way to Everest Base Camp, testing them on the way as we did in 2007. And I'm just going to show you a couple of interesting things which we think may explain the differences between us and them, certainly at some level. One of the things that we have been looking at in great detail now is something called microcirculatory blood flow. We have a small camera which you can put under your tongue and look at the way that blood flows through capillaries in the sublingual region. And I'm just going to cut to the results really for just for time's sake. One of the things we can do is measure blood flow. So already in 2007, we knew that as we ascend to high altitude, so lowlanders, the blood flow in the microcirculation gets slower and slower. Maybe that's because your hemoglobin is rising and the viscosity of the blood is increasing. We're not sure. And what you can see here in the black is the Sherpas and the gray is us lowlanders. Just as before, as the lowlanders ascend to Everest Base Camp, the blood flow slows down. Not only in the Sherpas does it not slow down, but it actually gets a bit faster. In terms of the density of the capillaries that we can measure with this device, we knew from 2007 that as lowlanders, our capillary density increases, and you can see that in the gray bars here, increasing from this level to this level. But you can see that the level of increase of capillary density is much higher in the Sherpas than it is in us. And the other measure that we can make with this camera device is we can look at the heterogeneity of flow in the microcirculation. So in most pathological conditions, the more heterogeneous the blood flow is, the more unwell a patient is. So you can see here, even at baseline and across, the lowlanders have very heterogeneous blood flow through their microcirculation, so some vessels flowing very fast and some vessels flowing very slow. But in the Sherpas, a much tighter control of blood flow through their microcirculation. And just to show you how obvious these changes are, this is the microcirculatory blood flow of a lowlander, and you can see that each one of these dots is a red blood cell flowing through capillaries, venules, and arterioles. And I think you can agree, and you don't really need to be much of a scientist to say the blood flow in this one is quite a lot faster. and that's generally how most of the Sherpa blood flow looks when you use this device under their tongue to examine it. And the last thing I'm really going to talk about is moving even further on than the capillary blood flow, and that's into the tissues and the mitochondria themselves. And we spent quite a lot of time now working with colleagues at the University of Cambridge understanding about how mitochondria work in the tissues. Unfortunately, to measure these values of mitochondrial metabolism and function, we can't get the information that we want from a blood test, which means that we have to have these biopsies. So we do an open surgical procedure and take out a small piece of skeletal muscle. We've all had many of these now and have these beautiful scars all the way down our leg as a sort of a badge of honor that we've participated in science. And we remove somewhere in the region of 150 to 200 milligrams of fresh skeletal muscle, dissect it down, freeze some of it, and use the rest of it as live tissue. In 2007, as I said, we only looked at ourselves and not the Sherpas, and these are the data that we published from 2007. The first thing to show is these electron micrographs that we work with Hans Hoppeler to produce, and the black staining on the micrographs, this is obviously skeletal muscle, but the black stain is mitochondria. And what you can see on the left is a normal amount of mitochondria in the skeletal muscle, and then after a prolonged stay at Everest Base Camp, after we've climbed the mountain, it's about a 35% reduction in the mitochondrial volume density of the tissue. So the amount of mitochondria has gone down substantially. When we then look at the function of the mitochondria in the tissue, there are significant decreases in complex 1 and complex 4 activity in the mitochondria, a reduction in uncoupling protein 3, which would suggest perhaps an increase in the efficiency of how the mitochondria are working. And this PGC1 alpha is basically a cofactor for mitochondrial biogenesis. And this may be related to the picture that you see here. So PGC1 alpha, usually if this decreases, you get less less production of mitochondria, hence this lower number here. What we've done since then is started using another technique. All of the work there, we basically freeze the sample, bring it home, look at in a laboratory. And in 2013, when we wanted to look at the Sherpas and how their mitochondria worked, we took these devices up to altitude with us. These are tissue respirometers, where you can put live tissue into these wells and then study the live tissue as it's functioning at altitude, just sort of fresh from the person that you've got it from. So we studied there both the Sherpas against ourselves. The Sherpas we studied in Kathmandu for their baseline. We found it a little too expensive to fly them all back to London for their baseline testing. And then on arrival at base camp, and just for us, a much longer time after we arrived at base camp. The process itself of this tissue respirometry, we basically put the tissue into the well, and there's a Clark electrode measuring oxygen consumption of the live tissue, and we add a series of substrates, uncouplers, and inhibitors to then look at oxidative phosphorylation and how it's working at the various stages. I'm not going to go into the details of the results because it takes quite a lot of time to talk through it. But the take-home message about the Sherpa mitochondria is that they work much more efficiently than ours. And part of the way that they do that is basically to stop using fatty acid or to reduce the amount of fatty acid that they use as a substrate for their mitochondria. Fatty acid takes a lot of oxygen to metabolize it to produce energy. And as a result of that, they have much higher energetics levels, so higher ATP levels in their tissue, and they also seem to have this sort of innate protection against the oxidative stress that is paradoxically caused by hypoxia. So we went away and thought about that for quite some time, and for us this was a bit of a revelation because all of us who have grown up as intensivists and anesthesiologists were always taught that the most important thing is oxygen delivery. And if you can do nothing else for a patient, just give them oxygen. As we are finding out from our clinical practice now, this may not be the case, and oxygen may in many circumstances be very harmful to a lot of the patients we look after. And for us as intensivists, it's made us think more and more now about whether oxygen consumption is really the answer to what's going wrong with our patients. So, and just to finish now, what we've done since then is that we've constructed a small but complicated project in the intensive care unit where we work in London, doing exactly the same thing as we did with the Sherpas and ourselves at altitude. So it was like a sort of bringing the Everest experiment to the intensive care unit. And we took these video micrographs again, looking at the microcirculation of very sick patients in an intensive care unit. We took muscle biopsies to look at mitochondrial function, and although I've not had time really to talk about it today, we took a lot of blood samples to look at nitric oxide activity. We've now finished this study and are in analyzing all the data from it. The mitochondrial data we've been looking at in great detail now. We used a control group, basically, so we got 21 critically ill patients, and what we did was to try and select the very sickest of patients that we could find on the intensive care unit with a 50% mortality. And then we had a small control group of age and sex matched patients undergoing hip surgery in our hospital. I can't tell you any of the results from these. We recently discussed the results at the meeting that Jaimo and I were at in Atacama. They're very complicated results from the mitochondrial function, but what I can say is that we found some very exciting differences between, firstly, both the controls and the intensive care patients in terms of mitochondrial function, and also between survivors and non-survivors in mitochondrial function, which tie in with the findings that we found in the Sherpas at high altitude. So to us, what we think we have achieved now is basically taken our intensive care unit to the mountain, understood what's going on inside us and the Sherpas on the mountain, and finally brought the whole story back to the intensive care unit. And very soon we hope to be able to publish these mitochondrial results along with the microcirculation and nitric oxide activity to really give you a picture of what we finally learnt about how we think the human body responds to hypoxia both at sea level and altitude. And with that, I think I'll stop, and I'm very happy to answer any questions. Thank you.